Siso-Fuertes, and today I'm just going to talk about the implementation of SIMVIS GEKKO in the clinical environment. So SIMVIS GEKKO is a binocular simultaneous vision simulator that was developed, is manufactured and commercialized by Twice Vision, which is the company that I'm currently working at as a clinical development optometrist. And SIMVIS GEKKO was developed, or visual simulators in general, were developed to overcome an unmet clinical need, because right now it can get difficult for clinicians to explain multifocal vision to patients. It's also tricky to manage their expectation. There is always a little bit of uncertainty, and there wasn't a proper screening tool until now. And likewise, for patients, it can get difficult to understand the concept of multifocality because it's quite a complex concept, and many of them are not even offered all the solutions that are out there. And obviously they couldn't try the lens they were gonna get implanted before implantation. So nowadays, most of the available visual simulators are based on adaptive optics and are available in labs, being perfect for R&D activities, and some of them are even commercialized. However, they usually have a large footprint. They are generally monocular and they have a small field of view. And most importantly, they are not see-through. They have a display that patients need to look at. However, SIMVIS GEKKO is a visual simulator which, with patients, can experience premium presbyopic corrections before actually undergoing refractive surgery. And SIMVIS GEKKO is wearable and is wireless. It's this device that you can see here in the picture. Is a headset that patients wear. It's programmable and is controlled with an iPad. So we can simulate multifocal intraocular lenses, multifocal contact lenses, even LASIK presbyopic patterns. The device has a wide field of view, 20 degrees or so, and it's see-through, so patients are seeing the real environment. It's not virtual reality, and there's not a display. It's the real thing. And the device is binocular, which allows clinicians to fine-tune the corrections as they wish. They can show patients monovision, mix and match, because they can simulate different corrections in each eye. So the main component of the SIMVIS GEKKO are tunable lenses, which are lenses that can change its power when a voltage is applied to them, and the image is projected onto the pupil plane of the patient. So when they work under what we call temporal multiplexing, there are fast periodic optical power variations that at speeds that are greater than the focus flicker fusion of the human eye, and this generates on the retina of the observer multifocal images that are apparently static. So in other words, here we have two foci, as we can see, the green and the blue one, and this happens at very, very high speed, and both images are superimposed, but for the observer, the appearance is static. So now you probably be thinking, how do we simulate real intraocular lenses? So we start by obtaining information about the lens design. Then we calculate how is the light distribution across the different foci. And then we calculate theoretically how much time the SIMVIS lens needs to stay in each of the foci in order to reproduce the same energy distribution. And finally, we evaluate this experimentally in an optical bench to make sure that the simulation is accurate. So the challenge after so many years of technological development was now to validate its clinical application and also to implement its use in the routine clinical practice, especially in the refractive surgery field. So one of the studies that was carried out and that has become the final step of our validations every time we introduce a new simulation into the device is the clinical validation, in which we measure the defocus scores of volunteer presbyopic patients wearing the SIMVIS GEKKO, and we compare those defocus scores with the focus scores reported in the literature from patients that had actually been implanted with the same intraocular lens. And these are the results that we obtained in a study where we included 15 subjects with clear crystalline lenses. And as you can see here, we have in color in orange, blue, and green the defocus scores measured in volunteer subjects with SIMVIS GEKKO. And in black dashed line, we have the defocus scores obtained from the literature from patients implanted. And as you can see, there's a high similarity between both the focus scores and indicating a very good correspondence of the simulation. And here we can see that the visual acuity at almost all the focus points from the SIMVIS GEKKO simulations are within the 90% of the confidence interval of the reported data from the implanted patients. But a question that many clinicians ask is, okay, this works with patients which has a clear crystalline lens, but what happens if my patient has a cataract? So here in these graphs, we are seeing the focus scores of patients who had the simulation done before undergoing surgery and they had cataracts, and the same patients were measured there the focus scores after surgery with the same lens implanted. So here we are not comparing with the literature, but the same patient pre and post-op. And as you can see, the safe similarities are also high, but the visual acuity was reduced overall preoperatively. And this is something that we've seen in several clinical studies. This is just an example. And it's explained because of the effect of the cataract. So the cataract is reducing the visual acuity, obviously. And to answer the question about the use of SIMVIS GEKKO in the presence of cataract further, another study was also carried out in which patients had to score different presbyopic corrections at different distances. So here we have far, intermediate, and near. And they did so before undergoing surgery, so in the presence of cataract, and also after undergoing surgery, but with the implantation of a monofocal intraocular lens. And as we can see here, the patients ranked very similarly the corrections before and after surgery, and there was also a high correlation of the scores pre and post-operatively, being the scores slightly higher, in the post-operative assessment, which makes also sense because the cataract had been removed. And here we asked a different pool of cataract patients to score different corrections at far in daylight and nightlight conditions. And as we can see here, that even in the absence of near vision assessment, which is probably the most meaningful assessment when we are simulating multifocal corrections, which provide very good near, we still can see differences between corrections very clearly in this graph. And we also gather feedback from this same pool of patients. And as you can see here, all of them agreed that it was really helpful to have the opportunity to experience multifocality before undergoing surgery. And they thought it was useful in helping them understanding the concept of multifocality. And they also agreed that the concept of SIMVIS was easy to understand for them. And a further step into the implementation of SIMVIS GEKKO in clinic implies providing clinicians with tools that help them in the assessment of whether a patient can accept multifocality or not, and also what could be the most suitable option for them. So this is because standard methods to assess multifocal visual quality could be visual acuity, the focus scores, questionnaires, and they don't measure really the multidimensionality of vision. They are usually time-consuming, and they also rely on patients' memory. So a new metric was developed, which is the multifocal acceptance score metric, or MAS for shorter, and it allows us to evaluate the multidimensionality of vision. So we ask patients to look at different images at far and at near, in daylight and in nightlight conditions, and we also measure stereopsis. And we get these types of polygons where in the center, it will be when patients score the images as a 0, so the worst possible, or in the periphery, we have the ones, the highest score, or 10, which is the best vision possible. So as you can see here, we can obtain different polygons for different corrections, and it gives us the opportunity to compare different corrections for the same patient, and also to ascertain which one may provide the best compromise for that particular patient requirements. And this is really important because here in this slide, we are seeing several mass polygons for all the subjects that were measured in this particular study. And what I want to highlight, because it's what stands out here, is that the same subject presents different polygon shapes for the different corrections, and there's no trend by correction across subjects. So if you look at the polygons with the same color, they don't have the same shape for all the subjects. So this means that there is a high inter-subject variability. So it seems relevant to give patients the opportunity to try different corrections before undergoing surgery, because one size may not fit all of them. And this is other outputs that we can extract from the mass metric. So we can analyze the visual compromise of presbyopic corrections. So we can see how far vision is degraded compared to a monofocal intraocular lens, and how the near vision is improved with a multifocal compared to a monofocal. So if we can integrate this metric, and we can measure stereo vision as well, and if we integrate all this together, we can generate an overall performance parameter that allows us to establish a ranking. So here, for example, for this subject, we have a ranking, and for another subject, we may have a different ranking. So the preferences are totally different. And in this slide, I'm showing the results of a study in which multifocal contact lenses were simulated with SIMVIS GEKKO, and the satisfaction was assessed and compared to the satisfaction and the performance in same patients, but with the contact lenses in place. So the questions were extracted from the CLUE questionnaire, and they were related to satisfaction at far distance and at near distance, both in day and night-light conditions, and also at night-light conditions for both far and near distance. And as we can see here, the answers were highly correlated with the mass score. So we can say that when patients answer a questionnaire right after fitting, their answers are highly correlated to the mass score. So just to conclude, so I just want to say that the focus scores of the fine vision panoptics and symphony intraocular lenses, which are the ones that I've presented in this talk specifically, simulated with SIMVIS GEKKO using public information, match perfectly the focus score from the implanted patients. Also, that cataract patients can benefit from SIMVIS GEKKO simulations preoperatively, and they are able to run different presbyopic corrections even through cataract opacification. And finally, that the multifocal acceptance score metric allows a rapid assessment of vision in a wide variety of visual scenarios that correlates very well with the results from questionnaires, and is therefore a suitable metric for use in clinic because it's highly repeatable, has a high sensitivity, and a short administration time. And thanks very much for listening, and I'll be happy to take any questions in the discussion time.